everyone, and a warm welcome to the final day of the 3-day international webinar on recent developments in biotechnology, hosted by the Biotechnology Department of St. Joseph's College of Engineering. Before we begin, I request the participants to adapt to the common instructions we have been asking to follow throughout the webinar. Submission of feedback forms is necessary to receive the certificate. The link will be posted sometime during the event. Lastly, I request you all to use the earphones for better quality. I now call upon the organizer, Dr. Justice Saikia Jacob, to deliver the welcome.
Good morning. It's my pleasure to welcome all in this third day webinar program. Respected professors, head of the departments, research scholars, and students from different universities in India and from various foreign universities, I wholeheartedly welcome you all for this webinar program. I also welcome our today's chief guest, Dr. Muralidharan, for accepting our invitation. Even though he is there in his hometown, he has accepted without any hesitation. Welcome, Dr. P. Muralidharan sir, for accepting our invitation. And I once again welcome all the participants on behalf of the Department of Biotechnology, St. Joseph's College of Engineering. Welcome. Thank you. Now it's over to the student organization.
Thank you for your kind words, sir. Our resource person for today's event is Dr. Pallayan Muralidharan. The topic of today's talk is alternative experimental models for screening. Dr. Pallayan Muralidharan is a well-known and experienced professor in the Department of Pharmacology at CL-based Mehta College of Pharmacy. He pursued his bachelor's and master's degree at JSS College of Pharmacy, Uti. He completed his PhD at Meenakshi Medical University, Chennai. He holds about 20 years of experience in the teaching field. Dr. Pallayan Muralidharan was given with a number of awards for his experience and dedication, such as the Best Teacher Award from Tamil Nadu Dr. NGR Medical University, Overseas Award from Biomedical Scientists Association, Unit Medical Preparation, and Lifetime Achievement Award in Pharmaceutical Sciences from College Academy and Scientific Society. With over 68 research published in national journals, 120 in international journals, and a total of 5 books have been published. He's more experienced in guiding students for projects which include 33 postgraduate, 30 undergraduate projects, and a supervisor for 7 PhD theses. He's a life member of Indian Pharmacology Society, Laboratory Scientists of India, and PETA. We are more than privileged to have you in this session today, sir. I now carry over the session to Dr. Pallayan Muralidharan.
So, good morning, everybody. And the topic for discussion today is alternative experimental models for drug discovery. Introduction. First, the drug discovery starts from identifying the chemical compounds and their nature by physical as well as chemical methods. Then, when it comes to the second stage, that is the preclinical drug screening. And here, the drug screening means it's a thorough investigation which is used to measure the pharmacological activity of a new chemical compound or a chemical substance, or else any combined chemical substances which is made for a purpose. And it is also to investigate the function of endogenous medications, endogenous mediators. In the sense, endogenous mediators are nothing but intracellular proteins which are used to enhance the activity and functions of other proteins, which is present in our body, and also to measure the drug toxicity and unwanted effects. So the use of animals, as you know, it's mainly used for food transportation, pets, and for sports and for other recreational purposes. And it's also used for championship and in as a old um, uses. These are all the old uses which we know. Everybody knows it as the human being itself. Whenever the human being started, it also started. And using of animals for the purpose of research is one of the extended uses. And in that, the drug testing and pharmacological as well as toxicological screening, which are useful in the development of new drugs for infectious and non-infectious disease, is the main purpose of animal use in the research. Then the species of animal used for screening, most of you might know, that is the mice and the rat. Apart from that, the xenophus frog, the rabbit, then hamster and guinea pigs, then the beagle, that is it's from the dog family, and cat, and finally the primates, which resemble the human gene to the maximum. And use of this experimental animals, we'll generally see what is the use of this experimental animals. So these animals serve as a tool to understand effectiveness of medical procedures and surgical experiments. And animals are used to test drugs like vaccines or to make drugs like vaccines, antibiotics, etc., which are used in diagnostics as well as treatment for humans. And the number of animals used in research have gone up with the advancement in the medical technology. So every year, millions of experimental animals are being used all over the world and killed. In UK alone, it is 4.75 million animals which were used for the research in the year 2019. And the total number of animals used in USA in the year 2019 was estimated to be 920,812 animals, while that in Germany it reached 4.13 million in 2019. So the volume of animals used were enormous. So the laws were implemented to control the use of these animals. Why? Because to prevent the pain, distress, and death experienced by the animals during the scientific experiments have been debating issue for a long time. So for to control that, the laws were used, and various acts and laws are there to bring the control over unethical use of animals and minimize the pain to animals during experimentation. And in 1824, that was the year the first law was used, and the Organization for Animals' Rights was formed by the Royal Society for the Prevention of Cruelty to Animals. And these are the various laws which was implemented in India. That is in the year 1960. The Prevention of Cruelty to Animals Act 1960 was amended, and it was amended in 1982. So the first act was in 1960. And then in 64, the Committee for the Purpose of Control and Supervision of Experiments on Animals, that is CPCSEA, was formed. That was in 1964. And then in 1972, it was modified into Wildlife Protection Act. And in 1992, the Indian National Science Academy, INSA, they formed the guidelines for care and use of animals in scientific research, and that was revised in 2001. And then in 1998, the and experiments, breeding and experiments on animals, that was amended. It was control and supervision rules in 1998, which was amended successively on 2001 as well as 2006. Then, in the year 2001, the Indian Council of Medical Research, that is ICMR, they framed the guidelines for use of laboratory animals in medical colleges. That was in 2001. And in 2009, the MCI amended the recommendations to use alternates to replace animal experiments. So they made a recommendation in 2009 to use alternative experiments. And in 2012, Ministry of Health and Family Welfare banned the use of animals in educational institutes. So only for the purpose of research they allowed. The other demonstrations and others were all banned in educational institutions starting from 2012 by the Ministry of Health and Family Welfare. And in 2013, the University Grants Commission set a guidelines for discontinuation of dissection and animal experimentation in zoology or life sciences in a phased manner. That was in 2013. So these were the laws which were followed and which are now followed for the alternative. Now the need for alternative models in drug discovery has started. Why? In laboratory, when you are using an animal, these animals can either be poisoned or it can be subjected to psychological stress or applied with the skin while it is applied with skin and eye irritants, and maybe it may be deprived of food, water, as well as sleep because of the psychological stress. It may not sleep. And another, these are the uh, problems faced in laboratory animals in the caring section. But in the experimental side, we have problems when you do experiments on animals. That is, pain, distress, and unethical behavior to the animals is there. And then requirement of skilled as well as trained manpower is necessary while we do the animal experiments. And the protocols might be time consuming and it may cost very high, and reproducibility of the result. The last point is very important here because when you do an animal experiment, it gives answer, one type of answer for us. When we are going for again, when we repeat the same experiment in another season, then the reproducibility of the result is not the same. There is a slight variations in the results. So these are the problems which we face when we use animal experiments. That is the need why we are changing for alternative models in drug discovery. And when we look into the matter of alternative models in drug discovery, there are three R's told by the people. That is reduction by the scientist earlier, reduction, refinement, and replacement in alternatives. So, alternative to animal testing was proposed to overcome some of the drawbacks associated with animal experiments and to avoid unethical procedures. A strategy of three R's is being applied, which stands for reduction, refinement, and replacement of laboratory use of animals. And the concept of replacement of animals was first discussed in 1957 by Charles Hume and William Russell at the University Federation for Animal Welfare. And they suggested some ways to make the animal experiments more humanly by their famous book, The Principles of Humane Experimental Techniques, and it was later called as 3 R's. So the 3 R's is this approach motivates the use of minimal number of animals, that is reduction in the total number of animals used in an experiment. And then the use of animals must be planned and it must be refined carefully in such a way that pain and distress caused during the experiment should be minimized. And any scientific method employed non-sentient materials which may replace use of conscious living vertebrates in animal experimentation is called refinement. What is that non-sentient animals is? It's nothing but they are also living animals like sponges, corals, and hydras, which don't have the sensation, but they have the living gene and other things. So we can employ any other non-sensient materials, that is replacement. And here the reduction refers to the minimization of animal numbers and refinement refers to the minimization of pain or stress or adverse effects and maximization of well-being over the course of animals lifetime. And a refinement provides a very good model to treat the disease and also minimize stress to the animals. And replacement refers to the replacement of animals on in various process. So we'll get into the topic like alternate animal models. The various methods have been suggested to avoid the animals use in experimentation. So the alternate means for drug testing is up to the preliminary levels only, and the advantages mainly they are much time saving and it requires less skilled persons and it needs also less space and it is much more cost effective when we follow the alternative methods. The first species available in the alternative method is Dan Rayero, which is otherwise called as commonly called as zebra fish, and it is a small freshwater fish with a length of 2 to 4 centimeter. And advantages of this model is it's nearly having a transparent body and which can be seen during the early development of the other features, and it easily helps for the visual access. So you can visually see the internal anatomy of the fish when it grows and the optical clarity of the internal organs shows us the we can directly observe the development stages involved in various organs. That is possible. And identification of phenotypic traits can be seen during mutagenesis and easy screening assessment of endpoint in toxicity testing is very clear and direct observation of gene expression is possible through light microscopy. And since the size is very small, short life cycle favors it much for the laboratory use. And the working space, cost of laboratory solutions, test chemicals, and the manpower involved are getting reduced when we use the zebrafish as an alternative to the animal experiments. And its embryos and larvae can be developed and used for testing in cell culture plates as well as petri dishes. and whole genome sequence is already available uh, as a source and makes this zebrafish as an attractive option for molecular as well as genetic research. So for this molecular and genetic research, since the genome sequence is already available, you can go for this type of research. And it has very less ethical problems in the experimental use since it is a lower vertebrate. And from infancy to the adult stage, it is used in a variety of applications. That is the first toxicological studies of chemicals and pharmaceuticals can be tried, and then anti-cancer studies, heart diseases, then neurological diseases, then behavioral diseases like Parkinson's and others, and Alzheimer's, and observe other mutations and problems in organ development due to exposure to the test molecules, and modeling of certain human diseases in zebrafish also can be done. And this one is used to ameliorate the disease phenotype and malfunctions in organ development. So zebrafish in scientific research. The zebrafish has been used as a model to understand the vertebral development and the neuronal basis of a behavior, that is ADHD, can also be studied, and to understand the human genetic disease like cancer. And it is also used for investigating the animal microbe interactions, how this animal interacts for a microbe in an environment, and to understand the gene function and human disease pathology, and also to screen the genetic pathway. This can be done. And for screening of drugs for its toxicity, this model can be used. Then about the another insect, it is fruit fly. It is Drosophila melangosta. So this Drosophila is a fruit fly. It is the common name, and it is being widely used uh, invertebrates, which is now in research. And it has a well-studied genome, which enables the study of molecular mechanism, which underlies the human diseases. And it has an advantage like 75% of the genes involved in human disease are believed to be same in Drosophila. And the fruit fly is of low cost and maintenance, propagation, and screening when compared to the mammal-based models. And results are very fast because this animal or the insect is having a short life cycle. The functions of various structures like heart, lungs, then gut, kidney, and reproductive tract are similar to that of mammals. And the response to drugs which are acting on CNS is similar to that observed in the mammals. Molecular and genetic tools have been made available to study this Drosophila, and due to similarities in developmental and behavioral activities, this fruit fly serves as a unique sensitive model for human genetics and diseases involved. And they have been used to study various diseases like Parkinson's disease, endocrine and memory dysfunctions disease, and muscle dystrophy, cell aging, programmed cell death, retrovirus biology, diabetes, and other toxicological testings can be done. And the benefits, such as a very short life cycle, small size, and simple anatomy, makes this Drosophila a well-known uh, tool to go for drug discovery. And large number of fruit flies can be used in studies for a single experiment result. So the statistical count also increases when you use this type of Drosophila, because many fruit flies can be used for a particular single experiment and can be caught within a short period with less ethical problems. So the ethical problems will be very less. Then the next substitute which we can use for replacement of animals is hydra. So this hydra is a small freshwater hydrozoan polyp which exhibits a low process of growing old. That is, it is multiplying very fastly, and the process of aging is very less in hydra, and it gives an very good regenerative and building capabilities throughout its life. That is, if you cut a part of hydra, the next day you can find that it has been regenerated. And this hydra shares 6,071 genes with humans, and it is better than this Drosophila, that is fruit fly, because it has only 5,696 genes in human, and it is truly immortal. So if you cut this polyp, the next day if you see, it must have replaced the damaged portion. So it is truly immortal. Hydra means it's truly immortal, and among 259 human aging genes, it was found that 207 were conserved in Hydra. And Hydra fulfills the conditions for providing a new potential model for aging studies, particularly. This Hydra can be used. And this Hydra offers a simple platform to study the effect of free radical biology. So it is being used to evaluate the toxic effects of bulk and uh, copper. So this nanoparticles toxicity can be evaluated using this hydra, and it is considered to be a good model to assess the toxic effects of water pollutants, and it is sensitive to xenobiotic as well as metal contaminants present in the medium. So it is a very good alternative to study about the environmental toxicology, and it is used as a screening model for teratogenicity and carcinogenicity studies. Then the other alternative is microorganisms. In that, the Saccharomyces cerevisiae, that is a brewing yeast, is most popular due to its rapid growth and ease of replicating and mutant isolation and dispersed cells. And this one is well defined. It has a well defined genetic system, and it has a highly versatile DNA transformation system. The generation time is very short, that is above 90 minutes, and hence it is very easy to grow a large population and use it for analyzing our studies. And it is best characterized genome maker, and one of the most ideal eukaryotic microorganisms for the biological studies, and presence of similar cellular architecture and same life cycle like multicellular eukaryotes is an advantage. And membranes bound organelles like nucleus, peroxime, mitochondria, and organelles, and secretory pathway also mimic the functions of this mammalian cells. And brewing is used to understand programmed cell death. So this alternate brewing yeast can be used for programmed cell death and cell death regulators in human and in cancer research. So mainly, this brewer's yeast can be used for cancer research, and it is also useful to understand the fundamental aspects of cellular biology in neurodegenerative diseases like Alzheimer's, Parkinson's, and Huntington disease. And by studying the endogenous or heterologous proteins that lay the root of this disease. So, this brewing yeast can be used for the study of neurodegenerative diseases. And then the computer aided drug discovery. This is structure based design, then ligand based design is there, which can be used for study of environmental toxicology, then metabolism, affinity, selectivity, and adverse reactions. So specified software programs are there to help or design a new molecule, and this is computer generated stimulations are used to predict various possible biological and toxic effects of a chemical or potential drug candidate without any animal dissection or without the use of animal dissection. So this promising molecules can be obtained from primary screening or used for in vivo experimentation. So first we'll screen with the specialized softwares available, and the softwares known as computer aided drug design, that is CAD. It is used to predict the receptor binding site for a potential drug molecule, and CAD works to identify probable binding site and hence avoids testing of unwanted chemicals having no biological activity. And such software programs can be used to make a new drug for the specific binding site receptors. So the examples part, that is, it is not available in this screen. I'll tell you. The software name is AMIDE, that is a medical image data examiner. The major use of this AMIDE is it can be used for medical image analysis in molecular imaging, and then the Xenogen living image software. And this one, software can be used for in vivo imaging display and analysis. And then for the behavioral study, we have a software called EthnoWatcher, which can be used for the behavioral analysis. And Mars is another software name, that is multimodal animal rotation system. This software can be used to study the animal activity, tracking the animal behavior and its enzyme activity and its drug delivery system. And this is the brief description with URLs which you can use for this computer aided drug design, that is PubChem, ChemBill, and then Zinc, ChemDB, ChemSpider. These are the various softwares used. And another tool is available in this computer aided system, that is structural activity relationship of computer programs. Here, if you see, that is quantitative structural activity relationship 3D and 2D QSAR. It is a mathematical relationship between the physiochemical properties of a drug molecule and its biological activity. Then carcinogenicity and mutagenicity of a potential drug can be predicted uh, by the computer database. And in the recent QSAR software tools show more appropriate results while predicting the carcinogenicity of a molecule. And the advantages of this computer models over conventional animal models are the speed and relatively inexpensive procedure. So these are the main points in for a researcher. And the cost of new techniques is very much less than the traditional laboratory practice. And then the next type of alternative, that is the cells and the tissue culture. So the use of in vitro cell and tissue cultures, which is involved in the growth of cells outside the body in laboratory environment, can be an important alternative, is an important alternative for animal experiments. And the cells and the tissues from the liver, kidney, brain, skin, etc., are removed from an animal and it is kept outside the body in a suitable growth medium for a few days to several months, or it can be preserved for years also. So you can grow the same cell in a medium. In vitro culture of this animal as well as human cells can also be done by isolation from each other and growing a monolayer over the surface of cultural plates or flakes or flasks. And cellular components like membrane fragments, cellular enzymes can also be used. And various types of cultures like cell culture, callus culture, and tissue culture and organ cultures are used for the screening purpose. Coming to the cells and tissue cultures. And benefits associated with this technique is easy to follow and less time consuming and are less expensive. And the main thing is it does not follow any ethical issues, and these methodologies are used for preliminary screening only of potential drug molecules and chemicals and to check the toxicity and efficacy. And almost all cosmetics which we use daily and drugs and chemicals are being tested for the toxicity and efficacy using this test nowadays. And for example, the eye retency test is being done on this tissue cultures only. And EU and ZU co-workers suggested an alternate which uses bovine corneal organ culture for testing the eye retency and for its toxicity. And to end the show, it is a summary that is Dan Trio zebrafish, which is mainly used for toxicological studies of chemicals and pharmaceuticals, and the Drosophila, that is the fruit fly, is used to study the Parkinson's disease mainly, and Hydra, this is used to evaluate the toxic effect of nanoparticles or environmental toxicity can be studied here. And then about the yeast, helps to understand the fundamental aspects of Huntington's disease, and computer aided models are used to predict the receptor binding site for potential drug molecules, and cells and tissue cultures used routinely for preliminary screening of potential drug molecules to check their toxicity and efficacy. So thank you. And in this juncture, I thank the management of St. Joseph's College for giving me this opportunity, and also for Mr. Justin for calling me to deliver a lecture. Thank you, one and all. Thank you.
Thank you so much, sir, for giving us detailed information on different animals used for drug discovery and issues faced by animals during study, and about 3 hours in experiments, followed by novel use of zebrafishes, hydra, and housefly as alternative models using various software techniques. I will now be reading out some of the questions our participants have asked in the chat box. Can tissue engineering models be used to replace animals?
Yes, we can replace the use of animals. We can take the tissues either from the animals for any particular, if an animal having a gene sequence, we can use that tissue for our further studies. We can store it for years together.
Okay, sir. The second question is, what about replacing every animal model with 3D cell culture tissue engineering?
Animal models like cell culture, we can use it to understand the fundamental aspects only. We cannot fully replace the cell culture technique for our cell lines for our study or discovery.
Okay, sir. Next question is about Hydra. So,
Mm.
Hydra conserves majority of aging-related genes on humans. If Hydra is immortal, can you elaborate on this?
Yeah, Hydra can be used for aging process studies. So it is mainly used for genomic studies only. Like the genes will be used for the study is mainly based on the genes, which is for aging. So if you are going to if you have a drug which is proven to be effective in other in vitro models, then we can go for testing this hydra because it is an immortal plant. Like so, once you test an chemical today, yeah, if it is an aging process or if it inhibits the aging, definitely there will be more growth or sprout in the hydra. And if it is a toxic one, the sprout or the growth or the polyp which grows the next day, there will be a reduction in the size of the polyp. This is how we can go for the evaluation of aging process using hydra.
Okay, sir. The next question is, what is the best animal or alternative model to study recombinant microbes as a vector to deliver therapeutic protein molecules?
We can use Drosophila for this studies because it's very easy and most
Oh,
of the gene and other mutations are all possible in the Drosophila model.
okay, sir. That's the end of the questionnaire session. And
Thank
I
you.
once again thank you, sir, for spending your valuable time and enriching us with information on various alternative animal handling techniques for conducting experiments and its advantages. Before we move on to the end of the session, I would like to let the participants know that the feedback link will be posted in some time and you have only 1 hour to fill it up. It is necessary to receive the e-certification. Now, I request our program advisor, Dr. Renuka Vishwanathan, to hold the valedictory function.
Good morning to all. I deem it a great honor and privilege to propose a vote of thanks on this occasion, international webinar on recent developments in biotechnology. Let me first of all start by giving glory to the Almighty for the smooth conduction of this event. First and foremost, I am grateful to the management, St. Joseph College of Engineering, St. Joseph Group of Institutions, Chairman Dr. V. Babu Manoharan Sir, Managing Director Mrs. Desipriya Madam, and Director Mr. Sasi Shekhar for giving us the opportunity and support in hosting this event. I thank our well-wishers, Principal Dr. V. Seshagiri Rao and Dean Dr. M. Parvatavardhani for their continued motivation. My heartfelt thanks to our HOD, Dr. G. Srikumar, for his valuable guidance and moral support, and the department faculty for their extended cooperation. With all sincerity, I wish to express my thanks to the speakers of this 3-day event, Mr. Ajay Punamakara, for his interesting presentation on chronic diseases and nutraceuticals, Mr. Selvaraj Arogyaraj, for his informative lecture on nanomaterial synthesis and their biomedical applications, and our today's speaker, Dr. Palayan Muralidharan, for his impressive session on alternative experimental models for drug discovery. My special thanks to the organizers, Dr. Justin Patia Jacob and Mrs. Yuvarani for their efforts in making this event a grand success. I would like to convey my thanks to all the participants, faculty, research scholars, and students from different colleges and universities and from various places for their evincing interest and great learning. Finally, heartfelt thanks to all the student coordinators who have worked hard by providing the technical and logistic support in making this 3-day webinar a wonderful success. Thank you.
Thank you so much, ma'am, for your thoughtful words. I gently remind you all once again to fill up the feedback form posted in the chat box. I thank all the participants for taking interest in today's event and also thank each one of you all for making this webinar series a great success by cooperating with us for the smooth sailing of this event. I hope to see you all in all the upcoming events hosted by the Department of Biotechnology, St. Joseph's College of Engineering. I end the session by saying satisfaction leads to stagnation, so never let your passion for growth ever die. Thank you and stay safe.